So chapter 33 talks about control systems in plants. And when we look at control systems, one thing we're going to look at is hormones. And in a soy plant, there are actually hormones in the plant that act similar to hormones in humans. So there's something called a phytoestrogen, and it's a class of plant hormones, but it acts on the estrogen receptors in humans. And so you can look at the health benefits of this if you're looking at women in menopause. Of course, too much estrogen can be dangerous. You can have cases where it may lead to breast cancer and other problems. So it's dose related, and of course, you could look at the difference between whether you want estrogen in men versus in women. Okay, the soy products contain isoflavonoids. These are the phytoestrogen that provide the human health benefits. Those are the estrogen acting compound that we saw on the previous slide. Experiments on how plants turn to light led to the discovery of plant hormones. And a plant is, this what plant is exhibiting something called phototropism, and that's light, and then tropism is turning or moving. So the plant turns towards the light. And you can see in this plant, obviously the sunlight was on the right-hand side of this plant. So when we look at how a plant actually turns, the way we think this is happening is the plants on the cells on the shady side, on this side over here, become elongated. They actually elongate these cells, and that causes the whole plant to turn toward the light source. All right. This was studied by Charles Darwin and his son, and what he did was he took grass seedlings and he planted them and put a light source onto one side. And his control was just a grass seedling. And then the one next to it, you can see he's taken the tip off here. And when you take the tip off, the seedling grows straight. And that tells us probably whatever is in the tip is controlling this phototropism. Now, if you cover the tip with something opaque, it also grows straight. If you cover the tip with something that's transparent, it will grow toward the light. So this is something that is light dependent here. If you cover the base, even with something opaque, it doesn't matter. It still will bend toward the light. So clearly this indicates the tip is probably the key. And then Boyson and Jensen, who had worked under Darwin and Darwin's son, continued this by taking off the tip and putting a piece of agar, gelatin here, through it. And whatever this chemical hormone was could pass through that, is what they discovered. And in fact, the plant would bend. But if they did it with a piece of mica, this active agent did not go through down to the plant. All right, so they were able to isolate this acting agent, and it was a hormone called auxin. All right, and it was determined that this is what caused phototropism. And the way it works is it promoted faster cell elongation on the shaded side of the shoot. So it actually acted on the shady side. And you can see in this experiment what they did to isolate it. They took the tip of the grass seedling and they put it on agar. And this hormone actually went into this agar. And then they discarded the tip and they just used the agar for this experiment. They took that and they stuck it across the whole seedling, and it caused the cells to elongate, and it went straight up faster than the control. If they put it on the right side, it caused the right cells to elongate, which caused the seedling to bend left. If they put it on the left side, the left cells elongated, causing the seedling to bend right. And if they put agar with no hormone in it, it had no effect. All right, there's 5 major types of hormones that regulate plant growth and development, and they will do that even in small amounts. And they can actually do different things depending on the concentration. One plant might increase growth in high concentrations and decrease it in low concentrations. They trigger what's called the signal transduction pathway that I'm sure you all remember. and they regulate plant growth and development. All right, here's the table of the 5 major types of hormones. This is also in your book if you don't feel like hitting pause and want to add it in later. You have auxins, which stimulate elongation, affect root growth, differentiation, branching, development of fruit, apical dominance, phototropism, and gravitropism. You have cytokinins, which affect root growth and differentiation. All right, they also stimulate cell division, and that's kind of their key thing is this cell division, stimulating cell division and growth. They stimulate germination and delay aging. Then you have gibberellins, also called gibs, which promote seed germination, bud development, stem elongation, and leaf growth. They stimulate flower and fruit development and affect root growth and differentiation. When we look at gibs and you look at fruit development, these are the ones that are sprayed onto your table grapes. When you eat table grapes, maybe you guys haven't noticed, but table grapes seem to keep getting bigger and bigger, and the little round Thompson seedless are now huge Thompson seedless. Just the basic grapes you eat at, you get from Safeway or whatever, are much bigger. They have been sprayed with gibs that affects the fruit development. Then there's one called abscisic acid, and there's ethylene gas, also called ethene gas in chemistry. Ethylene gas promotes fruit ripening. So as fruit ripens, this is the gas that is both released as it ripens, and it also promotes fruit ripening. So that's where we get that old saying, one bad apple spoils the bunch, because one apple that is very, very ripe produces ethylene gas, causing all the other apples around it to continue to ripen. Auxins stimulate the elongation of cells in young shoots. That's what we saw in those previous slides. One classic auxin, IAA, is the one that's from the apical meristem and at the tips of shoots. So when we talked about the dominance of the apical dominance before you cut off the top of a plant, it's auxins that control that. Okay, and auxins do different things depending on their concentration. So they can stimulate or inhibit the elongation of shoots and roots. And if you look on the graph on the right, you kind of get this sort of sweet spot for stems, which is right about here in 0.001 grams per liter and less for root development. And that's going to cause elongation. If you get too much as we move to the right, see auxin concentrations increase as we go to the right, it actually inhibits plant growth. Both stem and root growth get inhibited with the more auxin concentration you have. Okay, auxins may act by weakening cell walls. So this is a hypothesis, but it's a pretty good one. The way they think this works is you have auxin in your plant, and it's going to trigger a hydrogen pump that's going to lower the pH here, make it slightly more acidic. That's going to activate an enzyme that is going to break these crosslinks here. And then if you add water, essentially fill it with water, to the central vacuole, these crosslinks are now broken and the cell can elongate. Auxin promotes growth in stem diameter as well, and that's by stimulating the development of vascular tissue, your xylem and your phloem, and cell division in the vascular cambrium. And remember, that is the location where we add the new xylem and phloem. Cytokinins stimulate cell division. That's kind of their big thing. And they are produced by growing roots, embryos, and fruits, and they promote cell division. Cytokinins from roots may balance out the effect of auxins from the apical meristem. So remember this terminal bud that we were talking about, and if we chop it off, essentially, you get a bushier plant. Well, this bud here actually inhibits plant plant growth down here. And evolutionarily, this was probably sort of a race to the top for plants. It made them taller so they can reach up to the light. If they were in essentially competing for resources, they could grow taller faster. Gibs affect stem elongation and have numerous other effects. So on the right, you have a plant that has been treated, and on the left, you have a plant that has been untreated. Okay, here we go. Those beautiful giant Thompson seedless that you see everywhere. They stimulate the development of fruit. And so if you want bigger fruit for the same amount of time on the vine, you can just spray them. Great for table grapes. Not good for wine grapes because essentially what you have on the right side is just more water added to make them larger. You don't really have more flavor. It's mostly water. The abscisic acid inhibits many plant processes. It inhibits the germination of seeds. And so this is good when you're looking at seed germination and remaining dormant. It allows seeds to remain dormant until the environment is favorable, and then they can germinate. So this is, I think, the Mojave Desert, and this is after a flash flood. So a flash flood comes, basically washes away this hormone and allows all these plants to germinate essentially all at once to take advantage of the water that's present in the desert for a very short period of time. ABA also acts as a stress hormone. So if a plant is low on water and it's dehydrated, it'll help close the stomata, and that will prevent dehydration. Ethylene triggers fruit ripening and other aging processes. And so as fruit cells age, they give off ethylene, which causes the other cells to continue to age. So again, they both release it, and when they're around ethylene gas, they ripen. So here's a nice example of that, where you have ethylene triggering fruit ripening. So you have 3 bananas. These bananas were all yellow at exactly the same stage. The banana here in one was put in a bag with an orange. Now the banana releases a certain amount of ethylene gas itself, but the orange also releases ethylene gas. So this banana ripened faster than the one that was all by itself, just with its own ethylene gas. And then, of course, a beaker full of ethylene gas will release ethylene gas, causing the banana to ripen faster than if it was by itself. The falling of leaves is a changing ratio of auxin to ethylene. It's triggered by shorter days and probably causes the autumn color changes and loss of leaves from deciduous trees. So take a look on the right, and you can actually see a layer of tight cells right here. It's the purple layer. I'm actually a little to the left of where I am. That is a protective layer, so when this leaf and the stalk fall off, the plant has a little scar there that protects it from anything that wants to invade it. Plant hormones have many agricultural uses. Farmers use auxin. They also use gibs. Auxins used to promote fruit drop. So if you spray the right concentration, you can get the fruit to stay on the tree. If you spray the wrong concentration, you can actually make it drop faster by mistake. Auxin and gibs are also used to produce seedless fruits. Now, Thompson seedless is naturally seedless, but there are other fruits, including tomatoes, where you can make them seedless or fewer seeds, depending on what you're doing, by using hormones. There's also a synthetic auxin called 2,4-D, and that acts more on dicots versus monocots, so you can spray it on your grass, which is a monocot, and it'll kill the weeds, which are usually dicots. Tropism is the turning of a plant, and it'll orient the plant according to an environmental stimuli. Plants sense and respond to environmental changes. Tropins are growth responses. Obviously, a plant can't get up and move into the light if it wants to, so it's going to grow or change its shape to make it turn to the light. When we talk about response to light, that's phototropism, bending is a response to light. And remember, that was the result from auxins moving to the dark side of the stem. When we look at a response to gravity or gravitropism, that could be actually these little organelles that will settle to the bottom of the cells and allow it to orient. When you think about how does it know up from down when it's in the dark and it can't see, it's the settling of these little organelles that might trigger and distribute this hormone. You can see the plant on the left was left untouched. Here's a little seedling, shoot goes up, root goes down just like it's supposed to. This poor plant, they turned it 90 degrees mid-growth, and the stem was still able to figure out up. It was able to turn and go up, and the root was able to turn and go down. Plants also respond to touch, called thigmotropism. It's a response to touch, and when we talk about coiling of tendrils, this is a response to touch. You've seen, I'm sure, vines and other things that will coil around each other. How do they know which direction to coil? That is how they know. Plants also have internal clocks. An internal biological clock based off of 24 hours, just like humans when we talk about sleep and movements and daily cycles, your circadian rhythm, we're talking about a biological clock. All right, your innate biological rhythms and their fine-tuning, the circadian rhythms, what I'm talking about, it's about 24 hours even in the absence of environmental cues. And then it needs environmental cues to keep going with the synchronization, keeping it together day and night, right? So if you think about that, if a human gets on an airplane and they travel to Europe, they have jet lag because their circadian rhythm is jet lagged. It's for California time. And that'll last 24 hours or so, but then their internal clock will change to the new environmental cues of wherever they are. They don't always stay with California time forever and ever and ever. But for about 24 hours, our rhythm will stay the same. Plants are similar. You can fool them, but they have a circadian rhythm that lasts about 24 hours without any external cues. They could be in the dark. Okay, the nature of biological clocks may depend on the synthesis of a protein that regulates its own production through feedback control. What does that mean? It means when this protein has made enough of itself, when there's enough of this protein, it turns off its own transcription, which turns off its translation. It's not being made anymore. And as this protein is metabolized and the concentrations go down, it turns itself back on. Plants mark the season by measuring photoperiod. A photoperiod is the relative length of day and night. They use this for timing of flowering. It's also seasonal, right? The nights get longer in winter, the days get longer in summer. For a long time, people didn't know, like, how much was it temperature controlled. And temperature doesn't play a huge role in it. It can affect it a little bit, but it's almost always about the number of light hours. Okay, plants whose flowering is triggered by photoperiod fall into two groups. You have short day, which means long nights, right? If you have a short day, you have a long night. Or you have long day, which are short nights. And on the left, you can see the long day, the long day and the short night isn't flowering. So this one is a long, short day, long night flowering plant. But you can trick this plant by just doing a flash of light in the middle of the darkness, and it will think that it's 2 days, that it has 2 short nights. So a flash of light will actually trick this plant. You can also trick the long day plants, the short night plants, long day, short night, by making it think that there are 2 days here as well. Just by a short flash of light, you break up this long night, and it thinks that this is a day, and then this is another day. A phytochrome is a light detector that may help set the biological clock. And it's a protein with a light-absorbing component, and it's going to absorb light in either the red or the far red spectrum of the light spectrum. So it's either going to absorb red or far red. And this is what we think helps plants set their biological clocks. So if we look at plants, we can give them this red, a burst of red, which is like nighttime. But if you give a burst of far red immediately after that, we find the far red cancels out the red. So what they figured out was that this was one molecule, and it was just changing its, it was slightly altering its shape to either absorb either the red or the far red. So this is how this phytochrome works. It'll absorb one or the other, and it'll change back and forth. So here's the phytochrome. It's going to absorb red light. As soon as it absorbs the red light, especially during the day, it turns into a phytochrome that absorbs far red light. It'll also just turn itself back into this form in this slow conversion even in darkness. All right? But a red light will turn it this way, and a far red will turn the same molecule back the other way. So it's one molecule, just slightly different forms of it. All right, there's that lovely Arabidopsis as well, that wild mustard plant, and it was used to study hormones. Plant defenses against herbivores and infectious microbes have evolved in plants, and they have evolved in plants with the presence of these herbivores and infectious microbes. So some of them have worked out a relationship where the infectious microbes might still be there, but they don't harm the plant quite so much. And plants use chemicals very often to defend themselves, both from herbivores and from pathogens. So in this case, this is kind of cool. See that cute little caterpillar? Oh yeah, watch this. Poor caterpillar's about to get killed. So the caterpillar bites into this lovely leaf. So here we are. Caterpillar damages the leaf. The leaf goes been damaged, does a signal transduction pathway to create and release a chemical that calls the wasp. It's like a little signal. Hey wasp, look over here. There's a big fat yummy caterpillar here eating me. And the wasp comes, lays eggs in this sucker. It's alive while it does this. And then the larvae emerge and hatch from the now dead caterpillar. Okay, defenses against pathogens work like this. So you have both a local and a systemic defense against pathogens. So you have, here we have virus, and so it's attacking this leaf, and you can see the little yellow dots are the localized defense. In fact, these cells are going to kill themselves in defense. But it also creates a hormone that will go systemically throughout the plant so that the whole plant can make defensive chemicals. So that's the systemic part of it. The local part of it is a local chemical, and then the cells actually commit suicide. So your local defense includes microbe-killing chemicals and sealing off the infected area. The sealing off the infected area is the killing of the cells. The hormones trigger generalized defense responses in other organs, and that's what we call the systemic defense. Okay, and then plant biologists like to look at what animals do. So if we find animals out in nature that seem to medicate themselves, that's a clue for plant biologists that these plants might be useful in pharmaceuticals. So then they go, whatever you find animals medicating themselves with, we know to look there.